Hello and welcome to this seminar series. Today it is my great pleasure to introduce my colleague Era Taoufik. Era is a colleague at the Hellenic Pasteur Institute. She's a neuroimmunologist and she's been long interested in the study of cellular mechanisms underlying neurodegeneration and neuroinflammation in CNS pathologies. She obtained her first degree in London in the Royal Holloway University, and then her master's degree in molecular medicine at University College London again. And then she returned to Greece and she performed her PhD with Dr. Leslie Probert in our institute, at the Hellenic Pasteur Institute, where she studied TNF signaling. And in particularly, she was interested on how TNF and TNF receptor signaling is differentially used by neurons for neuroprotection, and how the neuron-immune interactions through NF-kappa B signaling modulated neuronal astrocytic and T-cell responses in multiple sclerosis and in stroke models, animal models. Then we became closer and we collaborated, and she became involved in the study of disease mechanisms using models of induced pluripotent stem cell based systems derived from patients with an inherited form of Parkinson's disease. But she also is using all the time transgenic animal models, and interestingly, she goes from the animal to the human system and back again. So this is an interesting sort of back and forth history. So Era today, she's going to talk to us about alpha-synuclein mediated synaptopathy at the core of Parkinson's disease and beyond. Era.
Okay, thanks, Rebecca, for the nice introduction. Let me share my screen. Can everyone see and hear me well?
Yeah.
Okay, thank you very much also, Vasu, for the kind invitation today. I was really looking forward to this. So today my talk is entitled Alpha-synuclein mediated synaptopathy at the core of Parkinson's disease and beyond. First of all, I just want to introduce, since we also probably have quite many students in the audience as well, some concepts before I go into showing our data from our team. And I'm going to start with the word synaptopathy, the term synaptopathy. And synaptopathy is used in order to describe diseases of the brain, the spinal cord, or the peripheral nervous system, that they are related to any kind of dysfunction at the synapse level. So in general, both neurodevelopmental disorders, but also neurological disorders are coming under the umbrella of this broader term. And usually the neurodevelopmental disorders are known as the main synaptopathies because we have multiple genes that they are linked directly at to the synapse, and they have been shown to be mutated and causing multiple neurodevelopmental disorders. On the other hand, it seems that synaptopathy in the late appearing neurological disorders, neurodegenerative diseases, is involved probably and is usually comes in our mind as the consequence of an ongoing neurodegeneration, and not so much as a causative effect that actually leads to these pathologies. So in general, and in this talk, we are actually thinking about synaptopathies and synaptopathy as a common denominator of all the neurological diseases. And you will understand also at the end of my talk why I'm introducing this concept here at the beginning. So before going though into synaptopathy, let's look a little bit at the healthy synapse. So this is a very, very simplistic overview of one healthy synapse that I chose to include in one review that we had in 2018. And there we can see very clearly that the CNS synapses are basically junctions between neurons, and they are there in order to transmit potentials, encoding information, and are largely divided in electrical and chemical, with the chemical actually transferring the activity unidirectionally from one neuron to the other through chemical mediators and neurotransmitters. So now, depending on the nature of these vesicles here, we largely subdivide them in excitatory and inhibitory, respectively. And throughout the mammalian brain, the dominant sources of excitation and inhibition are presented through glutamatergic and GABAergic synapses, as we can see here in this oversimplistic view that I decided to draw. More recently, though, another Taoufik with a Q, actually, published recently in PNAS, the protom of the synaptic vesicles in the mammalian brain, and this was actually in non-human primates and human samples, and they identified that this oversimplistic view is not simplistic at all. We actually have more than 700 proteins in the synaptic vesicles, and this actually is probably an underestimation. And these are involved in multiple functions, including the trafficking, the endocytosis of the vesicles, the cytoskeletal organization that is required in order to have proper synaptic function. Now, as I said, excitation and inhibition is there, and excitatory synapses are the most abundant, with the inhibitory being less abundant. However, it's very, very important to think of this excitation and inhibition as the yin-yang of our brain. And this balance is particularly important. And an in silico paper that I found, and it was really interesting, even though I understood little information from this paper. It's a systems approach paper that was published in Frontiers in Neuroscience in 2019. What happens is that it shows that there are synaptic connections, and the excitatory and inhibitory neurons are actually uniformly distributed throughout the system, throughout our brain. And they tend to form a network which is actually starting, and this is a simplistic network induced by a single initial connection between two neurons. So we can imagine that by changing one of these knobs, we can actually change the whole network, and we can actually change things that are happening both proximally, but also distally. And sometimes if these changes are at the core of this network, we can have a collapse of the whole system. In this paper published by Erin Schumann in eLife in 2020, it was particularly interesting to show, to see that we have multiple components of the synapse, both lying at the pre-synaptic terminal here, the postsynaptic density, and the adhesion molecules that come here in order to link this connection and stabilize it. And we now know, and based on this paper, that subtle changes, even changes in the half-life or minute changes in their levels, that they can be induced, for example, from polymorphisms or from mutations, they have detrimental effects both in the initial synapse formation and the maturation and establishment of the connectivity. So here comes Parkinson's disease, and Parkinson's disease actually is a common progressive neurodegenerative disease that I don't have to introduce to most of you, that results in the degeneration of the nigrostriatal dopaminergic pathway in the brain. The main areas affected are deep down in the brain, and the substantia nigra is, as you can see here, suffering from extensive neuronal loss. However, even though our mind always goes to the dopaminergic system, a few years ago, back in 2017, and after also having a talk with Professor Glamis, who I'm not seeing here, but maybe he's listening to us anyway, we started thinking that Parkinson's disease should be regarded not just as a dopaminergic failure, but as a system-level disorder. And we should start considering this pathology as something that lies not just in the basal ganglia, but outside as well, since we now know that we have a massive degeneration in other areas. It's an ongoing progression, and we also have areas that they are affected in PD, including the cortex, the cerebellum, and the hippocampus. So in general, neurological disorders, including PD, should be, from us as scientists, a system-level dysfunction, and should just, so in order not to focus on a specific pathway, which will aid us to look into the investigation of mechanisms in a more unbiased manner. Now, this system-level disorder is also evident at the clinical symptom level, and this is important because outside and except the motor symptoms that they do all arise from this dopaminergic loss, we also have a number of non-motor symptoms, including the loss of smell, autonomic dysfunction, cognitive decline, dementia, and psychotic features that are all associated with other brain regions and also are directly linked with excitatory and inhibitory signaling pathways in the brain. PD presents its form, it's presented in both sporadic, which is the majority, so almost about 90% of the cases are presented as sporadic forms. However, we also have the early onset forms that they are due to mutations in specific genes, that we have now almost 17 genes identified with specific mutations. However, in both the sporadic and familial forms, we do have aberrant neurotransmission. So what are these neurotransmission defects in Parkinson's disease? Except the well-known dopaminergic problem, it has been well established that we do also have glutamatergic neurotransmission problems, and these are directly linked to both motor and non-motor symptoms in Parkinson's disease. We also have a contribution of the GABAergic system, which is mainly associated with the non-motor manifestations, and these are also correlated with things that they happen quite early at the early stages of Parkinson's disease. And now we also have valuable information that shows that the serotonergic also system is impaired, heavily impaired, and it's also correlated directly to disability scoring in these patients. And the most important is that serotonergic system seems to be one of the very, very early systems to be affected, since it has been identified in familial Parkinson's disease patients when they are still asymptomatic, almost 20 years before they start having clinical symptomatology. Also, many things appear in many publications appear in the literature that they saw that the early degeneration could also be an interlink between dopaminergic and serotonergic, but also other systems as well. So this is quite evident now that we have multiple failure in multiple systems. What links all of this? All these are linked down to a small molecule, the alpha-synuclein, and alpha-synuclein is well presented in both sporadic and familial cases of PD. It's heavily loaded within structures which are the hallmark of Parkinson's disease pathology, the so-called Lewy bodies or Lewy neurites. And what is this protein? So this protein is quite small, it's 140 amino acid residues, and is predominantly and ubiquitously expressed in the brain, not just in the deep areas of the brain, but also in the cortex, the hippocampus, the olfactory bulb, and the cerebellum. So as a protein here, we can see that it has an N-terminal amphipathic repeat region. It also contains a hydrophobic and non-amyloid beta domain and a C-terminal acidic tail. And a number of mutations have been identified here, which are directly linked to familial Parkinson's disease. So this protein is also, as I said, heavily loaded within the structures, also in sporadic PD. But what do we know about its localization? So even though it was initially described as a nuclear protein, this is still under investigation. However, we know that this protein has a presynaptic localization as well, and this has been well established in animal models and in human tissues. So it's highly enriched in synaptic protons here that they sprout from the axons of different neurochemical phenotypes. So we can find alpha-synuclein in dopaminergic neurons, in GABAergic neurons, in glutamatergic neurons, serotonergic neurons. However, not all terminals tend to accumulate this protein upon pathology, and this again is something which is not, it's under investigation. However, what we know is that this protein is very, very important in multiple levels during vesicle endocytosis. It plays a role in the clustering, in the exocytosis towards the postsynaptic density, and the recycling of the vesicular pool. Now, alpha-synuclein, after many, many years of investigation, what we know about it is that there's a definite involvement in disease pathology, in PD and other also neurological disorders. However, the disease mechanisms today remain largely uncharacterized, despite the enormous efforts of multiple labs around the world. And the most important is that the physiological function of this small protein remains completely and utterly unknown. So our team here at the Hellenic Pasteur Institute has been interested in investigating the mechanisms of alpha-synuclein mediated neuronal dysfunction. And we have been focusing on a specific mutation for this reason, the PA53 alpha-synuclein mutation, which was the first actually mutation being identified in 1997, and which represents a missense mutation in the N-terminal domain of alpha-synuclein gene. So there are two main cohorts that they do carry this mutation. One is here in Greece, in the Peloponnese area, and another one in Sicily, which is known as the Contusi family. And this is a familial autosomal dominant PD that presents with severe clinical severity and also appears quite early and has a rapid progression. In addition, these patients, except their motor symptoms, they experience multiple cognitive symptoms and a rapid cognitive decline as well. Now, the experimental systems that we have and that we exploit in order to answer and address this question are both a mouse model, the M83 model that was developed a few years ago from the group of Virginia Lee and expresses the human A53 alpha-synuclein under the control of PRP promoter, so it's neuron specific. We also use these mice in order to isolate primary neurons. And also we have developed and characterized in our lab a human iPS-derived cellular model that I'm going to present you as well. So after 2 years of pandemic, I decided to kind of divide my talk, not in parts, but in seasons, like most of the Netflix series that we have been watching all this time. And during season 1, our lab has focused on the PA53 alpha-synuclein mediated synaptic dysfunction. We know that alpha-synuclein, the endogenous alpha-synuclein, tends to form monomers that they come together to form oligomeric structures that are heavily loaded at the spines, and these actually induce spine loss and reduction, leading to neurodegeneration probably. However, we have no idea whether the ongoing neurodegeneration is responsible for this process. Actually, whether mediated synaptic dysfunction from A53T, if it represents an early event or is the inevitable consequence of the ongoing neurodegeneration, it still remains the chicken egg or egg or chicken of in the synuclein field. So the season 1 started at the Human Stem Cell Unit at the Hellenic Pasteur Institute that was established from our team in 2012. In 2012. Georgina Kurupi, in collaboration back then also with Leonida Stefani's team, they actually developed a patient-derived disease model based on this mutation, where they actually used the dermal fibroblasts from patients carrying the PA53 mutation to genetically reprogram them and derive human pluripotent stem cells. These in turn were differentiated into dopaminergic neurons, and this system we have been using to identify disease pathogenesis, and as I will show you later, perform other tasks as well. So we actually performed, and what I'm going to show you is based on the analysis of 2 patient lines, of multiple lines from 2 patients and lines derived from 1 control. Here, very, very briefly, I'm going to show you, because all this has been already published by our group in 2017, that upon differentiation, we get a nice mixture of different subtypes of neurons. So around day 30 to between day 30 and day 50, we have dopaminergic neurons appearing, GABAergic and glutamatergic at similar percentages. And when we actually went on and looked at these neurons quite carefully, we've seen that they do contain alpha-synuclein aggregates. They do have extensive axonal pathology and degeneration. And this was very, very interesting because when we actually looked the original paper describing the post-mortem tissue from these patients, we also looked and we identified similar phenotypic neuropathological features. So this axonopathy, we also keep on seeing it when we actually compartmentalize the neurons using microfluidics. And we see here both human neurons and mouse hippocampal neurons carrying the 53D mutation, that they do have an impaired axonal compartment where less TUJ positive extensions are actually growing. So axonopathy is evident. We have extensive neural length problems as well, limitation, less neurites growing out of these neurons. So when we actually try to see what are the molecular mediators that they lead to this extensive axonopathy, the best way to go back then was to do a transcriptomics profile between PD and human and healthy neurons in the human system. So when we actually performed the RNA sequencing, it was quite interesting that out of almost 400 proteins, 72 belonged to synaptic signaling molecules. So when we actually tried to divide them into seeing which ones belong to the presynaptic, the adhesion, and the postsynaptic density, what we've seen is that we had an even distribution, and we had multiple defects with main downregulation at all parts of the synapse. So all pre-synaptic and postsynaptic, along with adhesion molecules, were significantly downregulated in this system. Of course, the next thing you think of doing is to go and compare, your data, your transcriptomics data, with PD data available that they're already out there. And when we did the comparison, we indeed identified quite a few of these molecules being overlapping with sporadic Parkinson's disease patients. However, to our surprise, is that most of these molecules were actually overlapping with molecules that they are either causative or directly linked to neurodevelopmental disorders, and mainly autism, or what we call actually, and we know as diseases of the synapse. So we had a significant overlap with autism, schizophrenia, and depression disorders, thinking that probably we have something that occurs quite early in this neuronal differentiation, and this is actually extensive synaptopathy in these neurons. This was also confirmed by neurohistochemical analysis, and we looked at the synaptic defects in PD neurons a little bit closer, and we identified that about 70 days to almost 100 days of cultures that they were growing on astrocytes in order to actually aid the synaptic connectivity, we had an impairment of synaptic connectivity. This was further validated using functional assays. So electrophysiology actually showed us that there was also an aberrant spontaneous synaptic activity in these neurons. And this was further confirmed when we actually did calcium imaging, where we noticed that there were again increased frequencies and amplitudes in these neurons at similar time points. Interestingly, when we tried, even though that was out of the scope of this paper, when we actually tried and transplanted these neurons in a model, which is a PD mouse model, after chemical induction by intrastriatal injection of 6-hydroxydopamine, what we actually observed is that these neurons, the grafts in the graft, they were having and they were showing a decreased capacity to form synaptic connections with the host cells, as we can see here. So overall, we had an idea that there is there are synaptic defects, and we also wanted to do this laborious task of trying to do electron microscopy in these neuron cultures That wasn't easy at all. However, with the valuable help of this mini Glucina at the Bioacademy of Athens, we managed to actually get some very, very nice pictures here, where we actually managed to see that not only we had multi-vesicular bodies, multilameral bodies, as you can see here, forming within these neurons, but also we had less or absent vesicles. So we had synapses that there were lacking vesicles, or some of them were looking fused. And we had in general an impaired organization of synaptic vesicular pools, as they were not even coming close to the presynaptic terminal. Further investigation using the electron microscope in the animal model, in the M83 mouse, actually confirmed these observations. And when we did the analysis in both cortical and hippocampal regions of 12-month-old mice, we also have similar observations, where we have diminished numbers of the synaptic vesicles compared to controls, where they were completely and utterly distinct, as we can see here. However, here we can see that they're completely and utterly sparse, and some of them were what we call the vacant synapses, because in the presynaptic terminal we couldn't see any synaptic vesicles. Now, so we have all these observations gathered, and we wanted to look at how early this dysfunction can be happening. And it was another challenging task that my student Elisabetta Acrioti pulled over, that she actually tried to see whether they are early synaptogenic defects in these PA53 neurons. So for this reason, we actually exploited and we modified an assay, which is called artificial synapse formation assay, where we actually co-culture neurons at different stages, and with cells that they are non-neuronal cells, like 293 cells, that they do overexpress a postsynaptic cue. So what we do here is we artificially give the signal to these neurons, and they actually come in contact, making an artificial connection that we can actually measure within 24 hours. So Elisabeth, also based on the work of Yang et al., she also improved this method in order to be able to measure with quite a strict way the synaptic connections that they are actually formed. Prior, though, to the artificial synapse formation, the first thing that she wanted to look at is how the excitatory, the number of excitatory synaptic connection and inhibitory connections looked in these cells. So when she used the mouse cells, that they do express the alpha-synuclein, she didn't actually, the mutant alpha-synuclein, she didn't observe any differences in the number of excitatory synaptic puncta. However, she observed a significant increase of the inhibitory synaptic connections. This was further confirmed when she went on to actually do the artificial synapse formation assay. And in this assay, she used two molecules. So she exploited in one assay NLGN4, neuroligin 4, which is the most well-established postsynaptic excitatory protein, and NLGN2, which is a postsynaptic inhibitory protein. She actually did the assay in both the human and the mouse system. And what she observed is that there was an increase of this artificial synaptic contacts in NLGN2 conditions. And this was also observed in the case of the mouse neurons. So overall, what we see is that we have an increased inhibitory artificial synaptic connections in the P53 neurons that correlated well with what we have seen as levels of synaptic puncta before. Now, in a parallel study that we also perform is that we look at the mouse, which is the M83 mouse. And as I said, this mouse has been used quite a lot, but it has been analyzed above 12 months of age, where it actually shows quite a severe phenotype. However, in the literature, we were lacking analysis that happens at earlier time points, and specifically analysis that had to do with neurotransmission. So we decided to actually compare the two time points, a late time point and an earlier time point, in order to identify whether they are differences in specific neurotransmitter molecules and their mediators. or transporters. So looking first at the dopaminergic system, what we identified is that the number of Th-positive neurons at different areas, because we looked at the nigra, the midbrain, and the hypothalamic areas, they were not actually, they were reduced at the 12-month-old mice, and there was a significant loss of Th neurons. However, this was not appearing at the early stage, showing that the decrease of dopaminergic neurons is limited at aged mice. When we went on, though, to look at the glutamatergic system by using GLUT2 and GLUT1, we didn't observe the same pattern. When we used GLUT2, we didn't observe any differences in any areas of the brain when we looked, and these are actually 12-month-old mice, but we had similar pictures coming from the earlier time point. However, when we looked at the VGLUT1, we identified that there was a significant decrease at an early time point, which also between 3 and 6 months old, and these mice were mainly around 4 months, in multiple areas of the brain, including the hippocampus and the cortex. And we had a clear reduction of VGLUT1 in M83 mice that preceded this identified in the dopaminergic. So overall, in this part, what we have actually done is that we have verified that there is indeed a synaptic dysfunction mediated by PA53T alpha-synuclein. And this is a schematic that we initially did when we derived our data from the RNA-seq by drawing a little bit of where we can actually position all these molecules that they are affected by alpha-synuclein. What we do know now, and based on our data, is coming both from the human neurons and the mouse model, is that we have a profound axonopathy. It happens at early stages of neuronal differentiation. We do have a concurrent reduction of multiple mRNAs that belong to synaptic molecules. There is aberrant synaptic connectivity and functionality, and also the glutamatergic system, at least in the mouse, seems to have alterations prior to dopaminergic neuron loss. Therefore, we know that there is a PA53 alpha-synuclein mediated synaptic imbalance that is still under further investigation to go further and analyze the exact mechanisms that lead to this severe synaptopathy. So, season 2 now. Since we were quite happy with how things went on season 1, we decided to go forward, since we had a system that had strong phenotypes that we could actually use in order to address the question of whether we can actually use the systems and try and reverse these neurodegenerative phenotypes. I'm including here a schematic showing that today, at least the most recent is 2020. This is something that I derived from clinicaltrials.gov. This is where we stand today with PD therapeutics. So most of the therapeutics that they are actually quite close to phase through are symptomatic. However, we have an emergence of disease-modifying therapies, and these are actually molecules also, including memantine and some other molecules here, that they do tend to target the synaptic imbalance that happens during these diseases. So our first approach was to target alpha-synuclein aggregation, of course, and we did this by obtaining compounds that they were targeting the process, the initial step of alpha-synuclein aggregation that requires basically binding of the N-terminal domain to lipid domains. So these molecules, they were derived from initially from UCSD. However, then they were bought by Europol. They were actually developed in the Eliza Maslaya laboratory in UCSD, and these molecules have already been shown in this original publication to be very effective in vivo mouse model of PD. Today, one of these molecules, MPT-118A, has successfully completed clinical trials and is actually currently ongoing into phase 3. because the phase 2 was just completed and it was also successful. So it's one of the molecules, one of the lead molecules in PD, and it was acquired by Novartis in December to be able to pull through and manage to do this large phase 3 clinical trial. So what did we see here? What we saw here basically is that we had in our model in the human neurons, when we added these molecules at quite early steps of the neuronal differentiation, that they were very, very efficient in rescuing neurodegeneration, as you can see here. But what was mainly important is that we had, and we could see it very clearly, that there were less aggregation of alpha-synuclein and a restoration of the neuronal connectivity, as we can see here from MAP2 staining. Now, most important is that not only we had a rescue of axonopathy, but we had the restoration of the synaptic connectivity. And here we actually exploited the system of monosynaptic tracing using the rabies virus that shows the connectivity ratio within a neuronal network. And what we have seen here is that the human A53 neurons, they have a significantly decreased connectivity ratio that was quite efficiently restored by the addition of this molecule. Furthermore, when we actually looked at the calcium dynamics, we've seen that the calcium dynamics, by looking at the amplitude and the frequency, again, we could see that there was a restoration using the NPT 118 molecule. So that was our approach when we actually tried to go and use a targeted strategy to target a specific part of the process. However, we also decided that since this was successful, it would be very interesting to see whether we could actually use the system to do a non-biased phenotypic screen. So our very talented and hardworking and persistent PhD student, Nasi Antonio, and in collaboration with the Pasteur Institute in Korea, she took the cells and she went and adapted them in the 384 plate in format in order to be able to screen a kinase inhibitor library. She actually managed to be very successful in this, and her readout was the TH levels and the TH density here. And from what she saw at the beginning is that she identified basically, and there were 4 hit molecules appearing in the screen that they could actually, as you can see here, reverse and improve the TH levels. When she came back, she actually did a dose response. She used all molecules, and she actually saw that the most promising molecule was this molecule here, BX795, that at specific concentration and most concentrations, it was inducing quite strong neuroprotective activity. So she went on and she actually performed a number of experiments where she actually verified that BX95 was particularly successful in improving the neurite length, the observed neurodegenerative phenotype.
Μας πιστεύαμε 2 και τις στέλναμε πίσω, ενώ τώρα στέλνουν 24, τις χρησιμοποιούμε όλες και στέλνουμε άδειο μπουκαλάκι. Γι' αυτό αλλάξανε και...
Συγγνώμη, Μαργαρίτα.
In addition, there was a significant increase on the aggregation process, and also the levels of the pathological phospho alpha-synuclein were also diminished. Of course, when you have a compound and it's a kinase inhibitor, and you know that okay, it works in a specific ways because we know that BX795 it can target specific kinases, including TPK1 and PDK1. However, it has been shown in the literature that it also has other multiple targets and also off-target effects. So the best way to go was again to exploit an omics approach, and here we decided to do proteomics since we have a kinase inhibitor, and we did this in collaboration with Martina Samyotaki and George Panagiotou at the Fleming Institute. And the proteomics analysis identified a specific cohort of 118 proteins that were restored after the addition of BX795. Interestingly here, we didn't see many proteins associated with the synapse, but we identified proteins that they had to do with core metabolic pathways, including RNA metabolism, protein synthesis, modification and transport, and also response to stress. Of course, when we look at all these proteins and when we are actually talking about protein synthesis, our minds go to mTOR signaling, and we wanted to see whether this compound affected the mTOR pathway or not. So we decided to focus on this, and since we also had some of these proteins, including RPS6, included in the proteomics, we decided to go there and focus on this, the analysis of this pathway. And indeed, what we saw is that BX795 was reducing phospho-RPS6. And as we can see also here, there was a significant reduction in these cells, suggesting that, showing us clearly that there was a definite mTOR involvement. So mTOR is there actually to induce protein synthesis and at the same time block autophagy. So one of the two main things that we wanted to see was the protein synthesis and whether it could have an effect on the overall protein synthesis. And this is something that we also quantified using an immunohistochemical-based assay. And we could identify that indeed BX795 was actually blocking phospho-RPS6, and indeed it was blocking the aberrant protein synthesis that we observed in this proteomics profile. Now, the compound actually, not only we have used our system, but we actually, in order to be sure and after, of course, valuable reviewers' comments, we obtained neurons from Fujifilm CDI. So this is another pair, and these are PA53 neurons that they are accompanied by their isogenic control. And we performed similar experiments in these dopaminergic neurons. That here we have enriched dopaminergic neurons in this culture, and we have seen similar effects that we have seen in our systems, including the diminishing of the aggregates, but also diminished levels of phospho-RPS6 and also of protein synthesis, verifying what we have seen as a neuroprotective effect of BX795. So overall, in season 2, the conclusions were that we do have a rescue of pathology when we actually target alpha-synuclein, but also when we actually use a kinase inhibitor that has not been previously associated this molecule with Parkinson's disease. And here we know that we have actually not just a molecule that has a very potent neuroprotective effect, but a key regulator of RNA and protein metabolism of P53 alpha-synuclein neurons. This work has been published recently, just now, I think, in NPJPD. So it actually, all this work yielded to this beautiful publication, but most important, what it gave us is that we actually realized that we do have a very powerful system in our hands, which is human-based, and we can use it for mechanistic studies to study PD pathogenesis, but also it's a valuable platform and a validated platform now in order to use it to identify disease-modifying compounds. So I'm moving to the upcoming season 3, which is actually just happening. And so we are very excited, and we are now addressing more challenges. And these challenges actually include something which has been particularly interesting to our team, to see how and when the PA53 alpha-synuclein-induced neurodegeneration and synaptic dysfunction initiates. So we do believe, and our data has supported, that this comprises an early event. Okay, but we do know whether it comprises an initiating event. At the same time, we also want to check whether PA53-mediated neurodegeneration can involve other cell types of CNS cells outside neurons. So where are we standing now? So our team, and basically Georgina, along with the help of our very good scientists here, Olivia, Georgia Thanasopoulou, and Katerina Seglia, what they have managed to do, first of all, they have developed the first human 3D organoid models of PA53 alpha-synucleinopathy. It wasn't an easy task because at the beginning they were trying to optimize the protocols, and this wasn't an easy job. So what Georgina did is actually used 2 protocols, and she combined knowledge from both in order to create brain floor plate NPCs at the beginning that upon a few days of differentiation, they tend to give what we call as midbrain organoids. That they tend to mature, and as you can see here, we keep them almost up to 70 days in order to characterize them throughout the maturation and growth process. So this is a timeline of their generation, and you can see here the distinct phases, as you can see also here what this distinct phase involves. It's very nice to see them in culture growing, and it's quite nice to see them when they are actually quite apparent in the dish. Here we have used an A53T line along with isogenic control in order to do all further comparisons that I'm going to show you. And at the beginning, we have actually stopped these organoids at day 15. And day 15 and also at day 30, these structures, they do contain numerous neuroepithelial rosette-like structures, as you can also see here. They are stained also with nestin, which is the marker for general neuronal stem cells, but also with FOXA2 and LMX1A, which are markers for the ventral floor plate cells. And what we have seen here is that at day 15, the percentage of the FOXA2 progenitors is comparable between control and PD organoids. However, MAP2 and TH neurons are much lower in P53T organoids. By going a few days later and stopping these structures at day 30, we observed that they do have the mutant organoids, they did have an increased FOXA2 cell composition compared to control, and the overall TH neurons gradually increased. However, they tended to remain reduced in the P53T case, as we can see down here. Now we are moving into later time points, 65 days and 95 days, and we still see here that FOXA2 and LMX1A progenitors tend to decline at these late stages, which is expected. However, what is important is that we have still lower TH neurons, that they do remain lower at even these late time points. What's happening with synuclein? Synuclein also is not apparent at the beginning. However, as neurons grow, it's more apparent and it gradually increases during differentiation. And what we can see here is that we have more apparent staining of alpha-synuclein in the neurites and extension of the neurons in the mutant organoids. Currently, Katerina is analyzing synaptic markers, and she's trying to establish a way to look at the synaptic connections and also address whether we have differences in excitatory and inhibitory synapses, but this is still ongoing, and we're just characterizing at different time points in order to be able to select the time point where we actually see very well defined localized puncta in order to perform further analysis. In addition, as I said, since we are now interested in how A53 can also impact the glial neuron interactions, and maybe neurodegeneration is not just cell autonomous, but it could have a contribution from resident glial cells as well. Floretia Papastefanaki, along with two newcomers in our lab, Christina Pasteur and Tassos Kolias, they have started to perform astrocyte differentiation. Astrocyte differentiation actually takes quite a long time, but they have managed to optimize a protocol based on the two previously published protocols in order to actually reduce the times that they actually derive these cells in order to be able to perform the analysis required in order to understand the contribution of astrocytes to neurodegeneration. So these are midbrain astrocytes. These are the first pictures of what we have. We characterize them with gold standards of astrocyte markers. And we can see that already in the cultures, we have different morphologies appearing, and they're quite distinct. They're much larger than the mouse cells, and they have much more distinct morphologies. And currently, what we have in the lab, and it's ongoing, is that we have tried to put them together, so neurons and astrocytes together in co-culture systems. They are viable, and they do so. They start showing some interactions, and these are still ongoing and in season 3. So as I'm concluding here, what we have done, I want to go back in my first slide when I actually talked about synaptopathy and synaptic dysfunction in neurodevelopmental and neurodegenerative diseases. Back in 2018, we published a review that was actually having this title here, with possible convergent disease mechanisms. And within this review, I was actually including the term that there might be a neurodevelopmental component in Parkinson's disease. So both reviewers back then, they asked me to remove this part because it was quite risky and intriguing, and there were not data supporting this notion back then. However, today I'm very happy because very recently in Cell Reports, the first paper by a collaborator of ours from Luxembourg, Jens van Borg, showed that there is actually, and it's also on the front page here, that another mutation in another gene in LRRK2, which is also responsible for familial PD, but also for sporadic cases, actually has a neurodevelopmental effect, and it probably affects neurodevelopmental pathways in such a subtle way, though, that it affects the neuronal populations that we end up with at a maturation level, which is more apparent at later stages of differentiation. So I'm going back to this original view, where we actually see the neurological disorders, irrelevant of whether they are early appearing or late appearing, whether mutations are causative or not, and I bring them under the umbrella of synaptic dysfunction, which is probably something that, at least in the neurodegenerative diseases, doesn't seem to be only the inevitable consequence of ongoing neurodegeneration, but it could be an early or even initiating event, which is now starting to get investigated. And of course, the single-cell transcriptomics are going to give us the valuable information on whether this is valid or not. So open questions in future directions. Many questions, of course. The more data you get, the more questions arise. We are very, very interested in understanding the underlying mechanisms that they lie under this, what we see as alpha-synuclein mediated synaptopathy, and we also perform now, there's ongoing collaboration with the Hellenic Institute, where we actually now perform this week our synaptosomal proteomics to be able to have a better idea of what happens at the synapse level. We are very, very interested to see and identify whether they are non-cell autonomous mechanisms of alpha-synuclein neurodegeneration and what the astrocytes, but also all other types like microglia do. And probably we will be able to also answer these questions in the 3D systems where we actually monitor the development very clearly in a complex and in an interactive cell-to-cell way. Of course, we are particularly interested, and this is something that is currently our focus, to understand and put a spatiotemporal pattern of the events of alpha-synuclein mediated synaptopathy. So we all know that it's happening. We don't know how early. We don't know whether it's a consequence or a cause. And we are currently organizing to perform single-cell RNA analysis in both our systems, mouse and human-based, in order to be able to put a spatiotemporal pattern in these processes. At the same time, we are particularly interested in by validating, by using our system to validate therapeutics that are mainly now targeting also the synaptic imbalance that we have observed in our system. This will be enabled by high content screening of compounds with both structural and functional restorative potential. And this will be actually enabled by two very important platforms that we're getting very soon in our institute, supported by a large grant rewarded to Rebecca Matsas from LIVEC for infrastructure. This contains a high content imaging system, and also a multi-electrode array, which will enable us to do high content screen also of electrophysiological parameters at the same time. In addition, this will also help us to add more compounds together. For example, use compounds that they target both aggregation, but also the synaptic imbalance, and see whether we have additive or combinatorial effects of multiple PD therapeutics. And I'm going to leave you with a photo from a virtual photo from our lab, since that was the most common that we have seen the last 2 years of the pandemic. We are very, very happy people, as you can see here. We are even more happier that we can actually have all these systems and all this potential to be able to answer and address these questions that I raised. Thank you very much, the Matsas group here at the HPI, for this fantastic work that we've been doing together all these years, and the environment that we have created. The Human Stem Cell Unit that now is led by Floridia Papastefanaki and is expanding fast. The Imaging Unit headed by Mika Thomaidou, and of course the valuable help for multiple things from Evangelia Xiki, and of course all our collaborators in and out of Greece, and of course our funding agencies that make all this work possible until today. Thank you very much for your attention, and session is open to questions.
Thank you very much, Era, for this extensive journey to the lab work. And now, please, you're asked to put your questions.
Shall I stop my share screen? Or shall I leave it just in case there are questions and I have to go back?
Can I start, Rebecca?
Of course, Spiro.
Era, it was an excellent presentation. Thank you very much.
Thank you.
It was too much, and I forgot most of my questions. But in any case, I want to ask you whether you observed in your in vitro systems or the in vivo systems aggregation of alpha-synuclein in neurons other than the dopaminergic.
Yeah, so I didn't really stress this out because it was also published in the paper of 2017 in PNAS. But interestingly, we have aggregation of alpha-synuclein in every neuronal type that we have looked. So we have in glutamatergic, in GABAergic, in dopaminergic, but also since these cultures are quite also immature, they also contain neurons that they haven't reached a subtype specification, but they are expressing MAP2 or TUJ. We also see aggregation in this more immature, let's say, neurons that they haven't acquired subtype specificity.
You said that that Parkinson's disease is not referring only to the neurodegeneration of dopaminergic neurons, but we have neurodegenerations in other systems. Do you think that there is also alpha-synuclein synaptopathy? And which are those other neurotransmitter systems that are being affected?
We know that the glutamatergic system is affected by alpha-synuclein. So there is literature showing at least a correlation, a direct relation between alpha-synuclein loads at the presynaptic terminal of, for example, of glutamatergic neurons. However, it's not clear. It's not clear still in the field because there is this peculiar thing with alpha-synuclein that even though it's a presynaptic molecule, it's not found always in all neurons, not in all areas. Even in glutamatergic neurons, it's not. It could be a neuron that has, for example, synuclein and a neighboring neuron that doesn't express it. So the field actually has not, let's say, clarified exactly the role of alpha-synuclein at the synapse. Okay. But there is a definite involvement, at least in the glutamatergic. And from what we see, if we actually try and correlate the data coming from functional MRI, which is now coming, more and more is coming out, is that multiple systems are affected. And when they go back and they look at postmortem tissues a little bit more carefully, and they start looking, for example, at serotonergic neurons, which is something that they did after the functional MRI and sporadic PD, they also saw that they are aggregates of alpha-synuclein serotonergic neurons. So we are still awaiting for the information to gather.
Thank you, Ira. Questions, more questions, perhaps?
Hi, Ira, thank you so much for the very nice talk. It was extremely interesting. And you have different approaches, co-cultures, in vivo systems, organoids, which was great, and drug screening as well. So I have a couple of questions, like most technical questions, like, for example, did you see in your organoids, even after day 90, any astrocytes?
First of all, we haven't kept them after day 90.
Okay.
We are at the process where we are actually, the last time point that we chose was day 90. We are now having more organoids developed that we are planning of keeping them for longer. Katerina just informed me yesterday that she has seen astrocytes at day 90.
Yeah,
so
that's what I would expect.
So they are there, but because they were, she just seen them and she didn't even have time to scan them at the confocal. I didn't have the opportunity to show them, but we are actually characterizing them now.
Okay, and the other thing, the other question is regarding also, since we mentioned about neurodegenerative diseases, but also it can be neurodevelopmental diseases. Have you seen any effect of these mutation in neurogenesis, like going back one stage before in the neural precursor states? Have you seen any differences in neural proliferation, either in monocultures or other in organoids, if you have seen anything?
So it seems that in the organoids, at least the precursors tend to be a bit more at the beginning, as I showed with FOXA2 and RMX1A. So this is a large part that I haven't shown today, since, first of all, there was no time, and there are many things that I wanted to focus on with the synapse, correlating with the synapse, is that we have also done transcriptomics analysis of our NPCs, and we have differences at the very early stages as well. So we have A53 mediated differences at the NPC level. And also when we do spontaneous differentiation of this human iPSAs, we also see some defects at the initial stages of neurogenesis, but these are still validated.
Okay, great. Yes, I want to see.
Rebecca, do you want to comment a little bit more on that?
I wanted to say that also the early phenotype that we see in the organoids where we have less neurons, okay, that might be a defect in neurogenesis. But I can also tell you that when Katerina analyzed the A53T mouse, she saw diminished neurogenesis in the dentate gyrus of the hippocampus. So there is a direct effect there of aberrant neurogenesis, let's say.
Good, thank you so much. One last question, because you mentioned about isogenic control cell lines. So at some point I was a little bit confused because you mentioned you used as a controller wild type isogenic line from a company.
So what happened is that our lines, we have two lines, wrong. We have multiple lines from two patients, and we also have multiple lines from one control that we are actually using routinely. We have tried multiple times. We've tried, I think, at least in 3 independent labs, to be able to derive isogenic controls, and we didn't succeed.
You mean with CRISPR-Cas9 to make? Yes.
We can't hear you, Ira.
Hello? Okay, now it's better.
Yeah, can you hear me now?
Okay.
Yeah.
Okay.
So in order to bypass the problem of the controls, because especially when you go into more mechanistic studies, you want to have isogenic controls. We obtained the lines. We obtained an A53T line along with this isogenic control from Fuji Film, CDC, which is a company that sells them. And we performed some validation, some of the validation experiments that we did in
Oh, we're losing you. We're losing you.
I haven't changed anything.
Okay. Now, so there is a loose connection.
Probably. I'm not moving. Okay. So also for the 3D organoids, we are trying to use the isogenic along with our lines, the isogenic pair, in order to be able, not to have the clonal variability between lines, and to be able to compensate for that as well, in order to get more substantial data and more validated data.
Okay, so you made you made it in the lab from fibroblasts the cell line, the iPS line with the mutation, but also the company has the same cell line.
It's from another patient, but it's the same mutation.
But the same mutation. Okay, thank you. That's why I want to clarify, because we were talking about isogenics and then isogenic line, and then I saw from the company, and I was a little bit confused. Thank you. Thank you very much. I don't have any other questions.
Any more questions from the audience? Okay, I don't see any raised hands or anybody else. So thank you very much, Era, and thank you all for being here with us. So see you next time in the next seminar.
Thank you very much.
Thank you very much, Era.
Thank you.
Thank you.
Thank you all for attending the HIAAD Basic Science seminar series.
Bye.
Ευχαριστώ, Βάσκο. Θα τα πούμε.
Βεβαίως.
Γεια σας.